Excellent. Hello, everyone. Thank you for taking the time to invest in today's webinar. We've got representation in the attendees from all over the continent. We've got from Vancouver to Toronto to Montreal, where I'm located, from Bay Area all the way to Boston. My name is Zishan Farouk. I am a senior account manager at Fluidigm. Quick housekeeping notes. At the end of the presentation will be about 40-45 minutes, around 11:45 a.m. Eastern. We'll open it up for a Q&A session. You'll be able to type your question in, and then we'll respond to as many questions as we can. For any that we don't get to, we'll definitely follow up with you. If a question comes in mind during the presentation, please type it up, send it in, so this way you don't forget about it, and then we'll make sure to get to it. All right, let's dive in. The technology being highlighted today is the Hyperion Tissue Imaging System. The application that we're going to focus on is Imaging Mass Cytometry. What makes today even more special is that it marks the official launch of the tissue imaging services offered from the Center for Advanced Single-Cell Analysis at SickKids Research Institute in Toronto, Ontario. Today's speakers will be Dr. Cynthia Gaidos and Eric Swanson. Dr. Gaidos is Senior Scientist, Developmental and Stem Cell Biology at the Hospital for Sick Children Research Institute. She is also a Professor of Immunology at the University of Toronto and Scientific Director at the Center for Advanced Single Cell Analysis at SickKids. Eric Swanson is a Senior Application Scientist at Fluidigm, and he specializes in imaging mass cytometry. He has been part of the R&D team that commercialized the Hyperion Tissue Imaging System. He has trained dozens of operators and helped countless more with technical and data analysis questions, facilitating some really cool publications. So again, thank you very much, everyone, for joining. Let's get started.
Good morning, everybody. I'm Cindy Gaidos. Very happy to be talking to you today, and especially like to thank those of you who are joining from the West. Hope you're snuggled in your home office in your pajamas with some coffee. It's a nice way to start the day. So as Ishan said, I'm really happy to introduce today the launch of our new Hyperion platform in our Center for Advanced Single Cell Analysis, which we call CASCA for short. As I think some of you know, we've been offering CyTOF suspension mass cytometry for many years now. And so some of you will be familiar with that. But we've recently added the Hyperion. And so what I'm going to do in the next 10 minutes or so is just very briefly introduce the technology and why we thought it was important. And I'm going to just show you a little vignette illustrating some biology that we've been using it to explore. So, uh-oh, now I can't seem to advance my slides. There we go. So as I said, I think most of you are familiar with mass cytometry and its advantages over conventional fluorescence-based flow cytometry, or imaging for that matter. And of course, even though there are flow cytometers available these days and fluorochromes available to be truly high parameter, the more colors you add, the more spreading error you have. And so while it's possible to use many fluorescence markers, the more markers, the more difficult it becomes. With CyTOF-based technology, we don't have that problem because we are using lanthanide metals tagged to antibodies. So we have the power of single atomic mass unit resolution and mass spectrometry to detect our antibodies and our markers. And so of course, this gives us power to have many more markers per assay in a mass cytometry assay, so that we can simultaneously measure all kinds of things: surface phenotype, survival, proliferation, transcription factors, cytokines, you name it. And the key, of course, shown over here on the right, is the ability to tag antibodies with polymers that have chelated lanthanide metals. So we've been using this technology, as I said, for many years. And I think one of the general areas of medical research and discovery-based biology that's benefited from high parameter is the area of immune oncology, the study of tumor immune interactions, both from again, a basic mechanistic standpoint and ever more therapeutic options these days. So I'm just going to give you a little brief vignette about how we've been using CyTOF in immune oncology. And this is a collaboration with Sean Egan, who's my colleague in developmental and stem cell biology, actually, and cell biology at SickKids. And he's been interested for many years in understanding the genetics of breast cancer. And this particular cancer I'll talk about today, type of breast cancer, is called invasive lobular carcinoma. And Sean is a really accomplished mouse modeler using really state-of-the-art genetic approaches to model human cancer in mice. And so he became interested in this particular disease, ILC for short, invasive lobular carcinoma. It is an estrogen receptor positive breast cancer, one of the rarer subtypes, so only 10 to 15% of all breast cancer cases. It has some very unique histological features, which are shown on the right. These include tumor cells invading stroma in what look like single file lines. There are two genetic markers of this particular class of breast cancer, which are loss of E-cadherin expression and activated PI3 kinase signaling. Other chromosomal abnormalities are common. And although women who develop ILC initially respond well to standard therapy, they suffer late relapses. So there is a need for new therapies for this disease. So as I said, Sean is a very accomplished mouse geneticist, and his lab created a mouse model of ILC by engineering mice that have floxed E-cadherin together with an inducibly activated form of PI3 kinase, and both those mutations are induced under the control of WAP-CRE, which is a breast epithelium-specific CRE that is turned on with lactation. And so on the right are just some slides showing that this particular form of breast cancer in mice mimics the histological features in women, including these single file infiltration types on the lower left plot and these round sort of targetoid kinds of structures. So Sean came to us, I guess about 3 years ago now. They'd already done a lot of work on this model. And then they ended up doing some bulk RNA sequencing and found very strong immune-related gene expression in these tumors, which is also a feature of a subset of human ILC, and it's called immune-related ILC. And so he came to me and said, Cindy, I know you do flow cytometry. Can you help us look at the cells in these breast tumors? And so we have a single cell quantitation of what kinds of immune cells are evading these tumors. So I said, Sean, of course we can do flow cytometry, but why would we when we can do mass cytometry and look at many more markers at once? So we had already been working on a broad immune phenotyping panel for mouse tissue. So this first panel shows you just a very high level overview of the CD45 positive cells in enzymatically digested tumors compared to control mammary glands, so mice without tumors. And this was mass cytometry, but of course it could have been flow cytometry. And we can see, if we just look broadly at the basic immune cell types in the tumor, there's no real difference between the abundance of these different immune subsets. But of course, because we had so many markers in our panel, I think this was a 33 marker panel, we were able to more deeply probe the kinds of immune cells in each subset, and that's where things got interesting. So I'm just going to show you a very brief summary, and you can read the paper that was published a couple of years ago if you want more details. So on this slide, I'm showing that these are T-SNE plots, which most of you will probably be familiar with, a dimensionality reduction technique for high parameter data. And we also use phenograph clustering to give us an unbiased view of the cell subsets in the control the control mammary gland on the top versus the tumor mammary glands on the bottom. And what really stood out initially was that there are CD11b positive cells in both cases, but they had very different phenotypes and clustered differently in the T-SNE space. So the mammary tumor macrophages are the two light blue clusters you can see on the top left, and those were present at lower abundance in the tumors, but replaced by another CD11b positive myeloid subset that we call tumor-associated macrophages because they had a unique phenotype, and cells like this have been seen in other studies. Notably, they expressed high levels of CD11c, integrin alpha 6, which is CD49F. So when we quantitate the clusters, again, we can see significantly that, that these unique tumor-associated macrophages are more abundant in the tumors. The heat map just shows, again, differential expression of several markers on the overall myeloid cell populations. And then what you can't really see because I haven't highlighted it, but buried in all of this data, we could also see some interesting differences in T cell subsets. First of all, the tumors shown in the dark gray boxes in the box plots had a lot more CD8 cells than the control mammary gland. But when we looked at the phenotype of those CD8 cells in the tumors, there were many more PD1 positive cells, so exhausted CD8 T cells, and the overall expression of PD1 was much higher on those CD8s. And when we looked at the CD8 negative fraction, there were also PD1 positive cells in the tumors, and including PD1 positive FOXP3 T regulatory cells. So lots of interesting tumor-associated differences in immune landscape. So that was interesting, and we published that, and of course Sean is going on to study other features of this model, including whether therapies targeting these exhausted T cells might present a viable therapy for this tumor. But in the meantime, we started to think about the spatial relationship of the cells in these tumors, because lots of data has emerged over the last several years in many tumor types, most notably melanoma initially, but also more recently in breast cancer, showing that the localization of immune cells within a tumor has information that can be prognostic and also predictive of responsive to immune therapy. So these cartoons show a couple of these examples. So on the left, there's a situation known as infiltrated, meaning there are immune cells in the tumor. It's not cold, but they're excluded. They're largely restricted to the margins of the tumor and don't infiltrate. In the middle, we have a picture showing an example of an inflamed tumor where the T cells and myeloid cells have actually infiltrated into the bulk of the tumor. And then there's this other interesting situation on the right that's known as infiltrated TLS, which stands for tertiary lymphoid structure. These are lymph node-like structures that sometimes can be seen in tumor margins, where there seems to be some organization of the immune cell subsets and collection. And these, again, can be prognostic in breast cancer in particular. So we thought we'd really like to take a look at where these different immune cell subsets were localized in our tumors. And of course, this is where Fluidigm's new Hyperion system to do imaging mass cytometry became very useful. So we installed this system a few years ago and have been working extensively to optimize it and get it ready for prime time. So I'm just going to show you a few examples from this study. So briefly, and Eric will talk much more about this in a few minutes, the Hyperion combines tissue sample laser ablation to the Helios mass cytometry platform. And the pictures show on the bottom left the slide scanner, which is you just insert your slide stained with metal tagged antibodies. And then there is a UV laser, very high power UV laser that ablates the tissue in slices. And these ablated pieces of tissue get sucked into the Helios to be analyzed by time of flight mass cytometry. And then computationally, the slices are reconstructed into a whole image. So we applied this technology to a couple of mammary tumors from this system. This is just an H&E of the first tumor that we profiled, which turned out to be an interesting one. In the red boxes are highlighted different kinds of histological areas of the tumor that we decided to profile, including these targetoid structures, these single file stromal kinds of structures, and then areas that looked like lymphoid accumulations on the margins. So possibly TLSs. In fact, I was concerned at first that we had dissected a lymph node with this tumor, but it turns out that's not what that is, as I'll show you. And this work was done by Michelle Zwo when she was a master's student in my lab, with a lot of help from Steve Morton-Toth, who's a master histologist in Jane Dansko's lab. Okay, so there's the 6 regions we profiled, just to put them all side by side. And what you're seeing on the top is the H&E stain, and on the bottom is the colored DNA stain of the tumor, so we can see that we sort of captured the right regions. So I'm just going to show you a couple of 2 and 3 color overlays to show how different the areas of the tumor are. So GP38 is a stromal cell marker, also on some endothelial, blood endothelial, lymphatic endothelial cells, and EpCAM is an epithelial marker. So we see that there are stromal and mixed epithelial cells in some of these regions, but that the regions we queried as possible TLSs were mostly devoid of both stroma and epithelial cells. Now, when we look at all hematopoietic cells expressing CD45 in the white, we can see in the top regions they are present, but they're largely excluded from the main body of the tumor. sometimes surrounding these interesting ductal structures. So these are part of the mammary gland biology that are marked by smooth muscle actin and CD31. And then there are lots of immune cells expressing CD45 in these TLS-like regions. So two macrophage markers that were in our panel, CD206, which is often considered to be an M2 macrophage marker, and CD64. And what you can see, what we hadn't really expected, was that these two macrophage markers are not really co-expressed. There seem to be regions of the tumor that have 206 positive macrophages and regions that have 64 positive macrophages. So probably two different kinds of macrophages in different regions. And lastly, the T cells and B cells. So in the general regions of the tumor, there's not a lot of T cells that have infiltrated in, but again, we can see small areas where there are starting to be accumulations of T and B cells around the tumor edge, including on the bottom left, this large lymph node-like structure that we're now calling a TLS, because the locations of the T and B cells within the structure don't resemble a normal lymph node at all. So there are really varying immune landscapes with respect to T and B cells in different regions of this tumor. And finally, we had another marker, which is CD2135, which is a marker of follicular dendritic cells, which are present in germinal centers. So these are very active B cell regions of an ongoing immune response. And so we can see that these follicles that had lots of B cells in the bottom left had very well developed FDC-like cells, and that they were also present in some of these other areas that where the structures were not quite so organized. So lastly, this is just a second tumor. I'll just show you a very quick summary of it, which showed many of the same features, and we focused again on these lymphoid or lymph node-like follicular accumulations of cells on the tumor margin. And again, we see very clear organization of these regions with different macrophages marked by CD206 in pink, and SIRP alpha this time in dark purple. They're in different regions. Within the lymphoid follicle in the top row, we can see that there are CD4 T cells, Tregs, and CD8 beta T cells, and that some of those T cells express PD-1. And then in the bottom region of the tumor, shown in the bottom image, again, we see sort of nascent developing immune-like or lymph node-like structures with various mixtures of T and B cells. And some areas, if you can see the small red accumulations where they're really only T cells. So really complex biology going on here that we can that we've seen nicely with this technology. And I'll just point out this latter work was also really aided by Mickey Gams when she was in my lab. She's now at Fluidigm, by the way, and Julietta, who was her summer student. So clearly, using this technology, we can see that there is a very distinct immune landscape that we can see that varies across different regions of the tumor. And again, based on other data in the literature, we think that this is really important to understand in detail. And those kinds of studies are continuing with Sean and I in collaboration to really try to better understand the immune significance of these localizations and how this might influence response to therapy. So I'll just acknowledge the people who did this work. So I mentioned Steve and Michelle in the beginning, and Mickey as well. I'd also like to shout out to Greg Chang, who was the manager of our facility for some years and has moved on into other directions recently, and his work has been taken over by Tina Chen, who has been with me for a long time and is now the overall manager of the facility, and will oversee launching of this platform. So thanks very much, and I'll turn it over to Eric now, who can give you a bit of a deeper dive into this technology.
So thanks, Sydney, for that great overview of the technology and some of the beautiful pictures that you showed. So today, during my talk, we'll focus on how you can, apply this technology to your research questions. And in particular, we'll look at how highly multiplexed immunohistochemistry and the simultaneous detection of up to 37 proteins in a fixed tissue section can occur with the Hyperion Imaging System. So a little bit of an outline. We'll start with introduction, then we'll switch gears and talk about some the workflow, user-generated data, how you can get started, and then last but not least, some support and contact information that you can use in your quest to get started. So without further ado, we'll start with a little bit of an introduction. As Sydney mentioned in her talk, right, the classic technology that people have been using for a long time is immunofluorescence. Immunofluorescence comes with a few different limitations. One, you have fluorescent spillover, right? So the peaks are very broad. They overlap into other peaks. You also have variable staining intensities between the fluorophores, right? So there can be a difference of 10 to the 3 fold change in, a very bright fluorophore versus a very dim. And last but not least, you have background signal, which often occurs from, autofluorescence or other endogenous fluorescent moieties inside your tissue. CyTOF technology and the Hyperion kind of attacks all these things because, one, we use very separate and distinct signals. So each isotope can be identified. The instrument has a 1 atomic mass unit resolution, so we can tell the difference between each metal isotope very easily. So our signals are very separate and they're distinct. We have uniform staining, right? So the difference between the peak sensitivity on the instrument and the sides of the range of detection is about 2 to 3 times, as opposed to that 10 to the 3rd as you would have for fluorophores. And last but not least, we do not have any background signal, right? So biological systems, mice, as Sydney was looking at, they aren't bags of, for example, ytterbium-176. We're composed of carbon, oxygen, nitrogens, and things like that, right? So all of the metals that are put into the system are put there because of the scientific perturbations that we're making in the system with, labeling and staining as accordingly. So what elements do we use? All of our isotopes, while they are mostly from this lanthanide series down here, which you probably forgot about in chemistry, they are also referred to as the rare earth elements. And turns out they aren't rare. They are quite common in the Earth's crust. They're non-biological, though, which is important for the applications you're using, because as I just mentioned, they aren't found in biological systems, and they're also safe to work with, right? They aren't radioactive. So you don't have to worry about any of those things. We currently have 135 different channels available, ranging from about arsenic to bismuth. But currently there's about 35 different metal tags that you can access out of the catalog. But many additional tags are possible, and academics and other groups have worked out novel chemistries that you can certainly use and leverage to increase the number of markers and gain additional information about your systems. So the Hyperion Imaging System, it is a dual-use platform, and Sydney hinted at this during her talk as well, right? It empowers multiple research applications. So you have the Helios, which is sitting here on the back in the purple box here, and that contains the plasma and the time of flight, and we'll go over that in just a little bit. And then out in front is the Hyperion, right? So the Hyperion Tissue Imager, it contains the laser for tissue ablation, as Sydney mentioned, and it's what we use to look at biological samples on a standard microscope slide. So, you can look at cells on a slide, you can look at tissues on a slide, anything that we can stick down to a slide that's biological in nature, we can assay with this instrumentation. All right, so what kind of things are people using the instrument for, and understanding about the instrument is one, it's very comprehensive, right? So you can do highly multiplexed immunohistochemistry using proven CyTOF technology that enables the analysis of up to 37 protein markers at a time. It's contextual, right? So you can identify new protein biomarkers and deeply profile that tissue microenvironment while preserving tissue architecture and cellular morphology. And last but not least, it's powerful, right? So you can interrogate multiple parameters from each section. You preserve those precious biopsy samples, and then you significantly reduce variability by eliminating that dependency on the serial sections that you would for traditional applications. It's also, being used for a wide range of questions, right? So people are looking at basic research questions, looking at stem cell differentiation, embryonic development, hematopoiesis. People are also looking at drug discovery and development questions, looking at different biomarkers, preclinical biological studies, and early development retrospective studies as well. And then finally, people are looking at clinical and translational research type studies and questions, right? Oncology, immunotherapy. They're looking at inflammation and autoimmune diseases. In Toronto in particular, there's been some very nice papers published on multiple sclerosis, as well as muscular dystrophy, stem cell therapies, and diabetes, among other examples. So, why do you need these, 37 markers? Why are people expanding their panels and wanting to use high parameters? And, the story I like to tell is that, it's needed to further the human condition and improve our medicine, right? So the current state of medicine, the top 10 highest grossing drugs in the United States, for example, only help between 1 in 25 and 1 in 4 of the people who take them. Consequently, there's a need for better design of clinical trials and identifying disease biomarkers that will help determine how patients will respond to treatment. And of course, this has been seen through a variety of different ways, right? For one, over the last few years, there's been almost 10,000 different clinical trials. And the probability of success with the biomarker versus without a biomarker increases by about 20%, right? And considering the cost of failed clinical trials anywhere from $800 to $1.4 billion, this is a big deal, right? And of course, that's reflected in the growth of immuno-oncology biomarkers that you've seen being published and looked at across, clinical trials that have concluded and are currently ongoing. And not only are high parameter biomarker analysis essential in improving the current state of medicine, but it's also important in understanding, spatial localization, right? So biomarker can also include the spatial localization and how those cells or the proteins might interact with other proteins in the surroundings. And a historical example of why spatial information is important that I like to talk about actually goes back to 1854 in London, and this was before they understood the germ theory, and there happened to be a cholera outbreak going on, right? And of course, if you look at a cholera outbreak in London, you could record the number of cases, you could record the number of deaths, a lot of different numbers that you could record. But John Snow had the idea that, well, maybe there's a spatial component to it, right? So because they didn't have an idea of how it spread at the time, he actually started plotting where the cases were coming from in London, and in particular around this Broad Street pump, right? And it turned out the cases and the number of cases were directly proportional to their proximity to the pump, right? And as we all know, cholera is a waterborne disease, so this makes a lot of sense, and it was very illuminating at the time. To take this into kind of a modern context, right, we now look at things like the proximity of cytotoxic T cells to the tumor and correlate that with patient outcomes and patient survival. And in fact, if you have a low number of T cells in relation to the tumor or a high number of T cells in relation to the tumor, you can see that your survival curves are quite different. But now if you add a little bit more complexity and a few additional markers, all of a sudden you can see that in this case, instead of just looking at T cells in their relation to the tumor, we're looking at CD3 positive, CD8 positive T cells and the distance of those cells to SOX10 positive or Ki67 negative cells. Now all of a sudden you can further stratify your patients, right? And really dive deep into that biology and understand what's going on. So this is one of the things that you can gain from multiplexing and adding those additional markers is you can further stratify your populations and understand the biology that much more. So how does this all work? How do you get started? How does the system work? Well, first of all, the first step is you have to design your panels, and we use immunohistochemistry validated antibodies conjugated to metal tags. We have a catalog of these antibodies available. You can also conjugate your own favorite markers, your own favorite antibodies, and Sydney's Core offers a very nice service that will do that for you. From there, you can stain, right? So you can stain your tissues, whether they be FFPE tissues, frozen tissues, fixed cells, basically anything that's placed on a standard microscope slide. And the protocols you use to stain your tissues are going to be remarkably similar to what you're doing for immunofluorescence today, right? The only major differences is you're going to need a strong thumb because you're going to pipette, 30 antibodies into the tube as opposed to maybe 2 or 3, right? And at the end of the staining, instead of using a DAPI marker to light up the nucleus, we'll use an iridium intercalator. But other than that, your staining protocols are going to be very identical to what you've traditionally been doing, and it's very easy to make that switch into this high parameter imaging on the Hyperion. Once you get your cells or your slides stained, you insert it into the Hyperion here, and then you image, right? And the image, as Sydney mentioned, is generated by a laser rastering across that tissue. And once you have, the image acquired, you're going to do your analysis, look at post-analytical, imaging, and use, probably some secondary analysis software and other tools that we'll discuss in a bit. So after the laser ablation, what happens? The little plume of material that's kicked up by the laser, blasting away at your tissue, gets carried into the helios, right? Here you have your inductively coupled plasma. This is running at about the temperature of the sun, about 5,000 degrees Kelvin. It ionizes everything with 100% ionization efficiency. From there, you go into the high-pass ion optic filters. So this filters out all those low molecular weight elements, right? Those biological elements, your carbons, your nitrogens, your oxygens, your sulfurs, right? Leaving behind those high-weight rare earth elements that you labeled your system with, right? And then, of course, your masses are separated by time of flight, where the lighter ions hit the detector first, the heavier ions hit the detector later. And what's really cool about the technology, right, is, depending on the number of ions that hit the detector, for a given plume of material, right? You have a peak, and this peak correlates directly to your signal intensity, right? So if we look at this red image here, where you have pixels that have a bright red color, of course, this red peak is going to be higher than in pixels where you have the black color, right? So, the intensity of the pixel and of the spot in your image that you see is directly correlated to the number of ions that hit the detector for that given pixel, right? So the data that comes off of this is very quantitative. From there, you will generally do some cell segmentation algorithms to pull out single cell data. And then from there, people will look at, proximity, what cells are bordering the given cell. They'll do some dimensional reduction and other downstream data analysis to delve into their biology. So how does, the Hyperion compare to immunofluorescence? This is actually images taken from the exact same tissue section, the exact same region, right? On the top, you have immunofluorescence done with some confocal microscopy, and this is courtesy of Bernd Bodenmiller's group at the University of Zurich. And one of the things I'd like to point out is, besides the images looking very similar here from what you have on the confocal compared to what you have on the Hyperion, on the confocal, you actually have regions of autofluorescence, as you can see here in this HER2 staining. But on the Hyperion Imaging System, that autofluorescence is missing, right? And it's gone, right? And that's simply because we don't have to deal with autofluorescence, as I mentioned previously. But, it's important to remember that what you're going to get out image-wise from the Hyperion, it's going to be very similar to what you'd get with, somewhere in between a 10 to 20x objective on your confocal microscope. Also, other things to keep in mind, what the Hyperion speed and throughput is, right? The imaging speed, it's dependent on the laser frequency. We're running at 200 hertz, which means we have 200 laser shots per second. And each laser shot ablates a 1 micron by 1 micron square area of tissue, right? So imaging speed is also independent of the number of markers, right? So all staining is performed at the same time, and all of your up to 135 different channels can be acquired simultaneously. Your example throughput per day, just to get this into kind of a more meaningful information, would be about 12 millimeters squared of tissue, which translates into about 45 TMA cores if they're 0.6 millimeters across, or 15 TMA cores at 1 millimeter across, right? So you can get 15 1 millimeter by 1 millimeter areas over 24 hours of operation. And one of the very nice things about the instrument, it is very set it and forget it, right? So you can set up, 45 different cores to be acquired on Friday afternoon and come back, Monday morning to a huge wealth of data that's available for you to look at. Right, and that kind of hints on this last point is, multiple sections on one slide increase the throughput because the only, human interaction you need with the instrument after you push go is taking slides in and out. and setting up a new acquisition parameter. So current use of imaging mass cytometry, we have over 80 instruments worldwide now, North America, Europe, Asia, 50 plus publications, right? And these numbers are growing, you know, every day, right? And then we also have 4 different clinical trials occurring across the world right now. These are phase 1, phase 2, phase 3 clinical trials. One's looking at vaccine development, which is quite prevalent right now, and others are looking at breast cancer and glioblastoma models. So, you know, one example I like to give that's available in a white paper that I can share is a study that we did internally looking at lung adenocarcinoma tissue, right? So here we had 20 different markers, right? Staying with a 20 different marker panel. And what's really nice about the technology is after you do your single cell analysis and you segment out your cells, right? You can plot your cells here using a t-SNE plot. And for example, here you can, identify different populations in your images, right? So here's your immune cell population up here in blue, your epithelial cells here in red, your cancerous cells here in green, and then stromal cells here in this, navy color. And what's cool then is you can then take, for example, here your immune cells and then highlight them back onto your image, right? So you know where these cells are coming from in your image, and you can do this round trip kind of analysis and look at things and assess, are your immune cells inside the tumor? Are they outside the tumor? Where are they in relation to the blood vessel, right? And all of this can be done and analyzed with open source software that's available that Fluidigm can train you how to use. So now we'll actually look at some user-generated data, right? The first example I have to talk about today was a paper that came out recently looking at smooth muscle cell reprogramming in aortic aneurysms. And, I love this paper because it's very visual, and the authors really told a very nice story that's, cool to think about. So, based on bulk RNA-seq samples from human patients with aneurysms from human patients with aneurysms, the authors discovered that there's an inhibition of smooth muscle cell TGF-beta signaling, and accordingly, they created a mouse model, right? And they put this mouse model on an ApoE knockout background with a tamoxifen-induced deletion of the TGF-beta receptor in smooth muscle cells. Right, and as you can see, these knockout mouse models, had huge amounts of lipid accumulation compared to the wild types, and they also experienced, very nasty, I guess would be a good word to look at it, formations of aneurysms in their aortas. And when the authors then went to image these aortas, they realized that here there's a GFP reporter highlighting the smooth muscle cells, right? So you can see those in green. And in the ApoE knockout mice, you see high expressions of this H11 marker here, right? So that's co-localizing quite nicely with the smooth muscle cells expressing that green GFP. And of course, this is all done in the Hyperion too, right? So this is secondary looking at the GFP. But independent of that, you can see that this smooth muscle cell protein is kind of absent in these aortic nodules that are forming and are the cause of these aneurysms, right? And what markers are present in these nodules are markers like aggrecan, osteoponectin, and adiponectin, right? So the authors found that these markers are actually characteristic of a mesenchymal stem cell type lineage, right? And when they did their cell segmentation and looked at the single cells and did their dimensional reduction, they found that these population here, 19, right, which was only found in the knockout mice, expressed, this OPN, aggrecan, GFP, and adiponectin, right? So obviously there was some kind of transition from that smooth muscle cell to this mesenchymal stem cell cell type, right? And this correlates with the hardening that you see in these aortic aneurysms and some other data that the authors published here that I'm not going to discuss today. And what's more is, when you start looking at, kind of classical mesenchymal stem cell markers, right? The authors noted that there was an increase from 1 month, which I don't show that data here, to 4 months, which are what these pictures are here, in these lesions, right? And you can see the upregulation of many mesenchymal stem cell markers, in the knockout mice, including, CD105, CD73, CD90, and CD44. Right, and these cells that are expressing these markers are clustering quite nicely around these nodules, right, when they do their cell segmentation and single cell analysis of their images. Right, so from this work, the authors were able to conclude that TGF-beta signaling inhibition and high cholesterol is needed in combination to promote smooth muscle cell reprogramming. The reprogrammed smooth muscle cells express many markers of mesenchymal stem cells and are found clustered together in mouse aortic aneurysms. And then multiplexing allowed for the imaging of smooth muscle cell to mesenchymal stem cell transition. And the visualization of mesenchymal-derived cell types included, adipocyte, chondrocyte, osteoblast, macrophages, and all of this was done, in a single tissue section, which is quite cool. And I should also add that, all of this analysis is done using open source, readily available tools that Fluidigm can help you use and teach you how to use to, attack and address your own research questions. The next example I like to talk about is a recent paper that came out looking at cancer-associated fibroblasts and how they promote immunosuppression by inducing our reactive oxygen species generating monocytic MDSCs in lung squamous cell carcinomas. So here, the authors first examined lung smooth cell carcinoma transcriptomic data for expression levels of fibroblasts, for expression levels of fibroblast activated protein, or FAP, and tumor infiltrating immune cell markers. And high correlations were observed in their RNA-seq data, between CAFs, right, and monocytic myeloid cell markers, including CD14, CD16, and CD33. So they wanted to validate this using imaging, right? And consequently, the authors performed IMC on 10 primary lung smooth cell carcinoma tumors using 17 markers to identify fibroblasts and different immune cell populations. And what the authors found is that high levels of FAP positive CAFs correlated with high levels of CD14 positive myeloid cells, and phenotypic analysis of the data. Phenotypic analysis of the data showed several clusters, including here clusters 5, 6, and 7, 5, 6, and 12, with high FAP, smooth muscle actin, collagen, and CD9 expression, which is indicative of CAFs, right? So here you can see cluster 6, it's expressing smooth muscle actin, it's expressing fomentin, it's expressing FAP, right? And when you look at the neighborhood analysis of this data, right, you can see that these CAF fibroblast cell types were also interacting with monocytic cell types, right, which in this case would be cell phenotype 4, right? So here, cell phenotype 4 is characterized by high expressions of CD14, CD16, right, and other markers of monocytes. And when you look at this clustergram here, you can see that cluster 5 is also interacting quite nicely with cluster 4. Right. So these CAFs were in spatial proximity to the monocytes, and also interestingly, in these same tumors, the authors realized that there was also this cluster 1 which expressed CD4 and CD3. In addition to, express CD4 and CD3, which, is characteristic of CD4 positive, CD25 positive T cells, which, create this immunosuppressive environment, right? And it turns out that these patients ended up doing poorly. And, when the authors then did some subsequent studies looking at cell culture and other models, they found that they could correct this tumor suppressive environment in the tissues. right? So what the authors were able to conclude from this study was that one, IMC revealed high levels of interactions between CAFs and monocytes in lung smooth cell carcinomas. CAFs also interacted with CD4 positive clusters in line with findings showing that FAP high, PDGFR beta high CAFs recruit CD4 positive, CD25 positive T cells to the tumor microenvironment. And correlation between CAFs and other immune cells was not significant. And these results confirmed what was observed in transcriptomic data. And the interaction between CAFs and monocytes can be inhibited by disrupting the CCL2, CCR2 signaling axis or inhibiting ROS generation. And targeting these pathways may provide novel avenues for cancer treatment down the road. And then the final example of some customer generated data that I like to present and go over is pancreatic islet profiling that was published in two separate diabetes, looking at type 1 diabetes patients that came out a few years ago. And one of the reasons I like highlighting this study is because, it's very hard to get your hands on type 1 diabetes tissue. And here you can preserve that tissue by looking at all the different types of, cells that comprise a pancreatic islet, right? So you can see here in this image that the authors are looking at insulin, glucagon, pancreatic polypeptide, and at the same time, which isn't shown here, but they also had in their panel, they also had many markers looking at different immune cell infiltrates, so that they could really discern what's going on in these type 1 diabetes islets. So now that you've seen kind of some of the questions that people have asked and answered and some of the published results, how do you get started, right? How do you design your panels? How do you do some image acquisition and data analysis? So panel design, you have 100 different max par conjugated antibodies for FFP tissue. And when we say that these are verified antibodies, they've been verified by a pathologist, outside pathologist, independent pathologist, that was working with us at the time. And we also have 40 plus IMC verified antibodies for frozen tissue, and new antibodies are added to the catalog on a rolling basis. Right. We also have max par conjugation kits. So if your antibodies aren't present in the catalog, you can conjugate your antibody to your metal of choice. Protocol is straightforward, takes about 6 hours. Multiple antibodies can be conjugated simultaneously. And the big caveat here is that your antibodies must be in a carrier-free protein formulation. So what does this IMC verified antibody mean? Well, you can look at the data sheets. They're all published on our website. And the pathologist, when verifying the antibodies for us, they look at expected patterns of co-localization, counter-localization, expected positive and negative staining patterns, as well as co- and counter-localization as well. We also have some ready-to-go MaxPar human oncology panel kits. These include the Iridium DNA intercalator, and there's an application note that's available to look at these. But basically, there's 3 different flavors of kits, and these kits can all be combined and mixed and matched to easily expand and customize your panels, right? But we have a tumor-infiltrating lymphocyte kit, an immune activation kit, and a tissue architecture kit. And these are for FFP tissues. Here you can see them in action, right? We have, this is looking at human breast cancer tissue, right? And you can see different populations of cells that are being lit up quite nicely by all of these markers. But it's important when it comes to panel design that you do want to include some tissue relevant structural markers because it's very useful for segmenting your tissue. In this case, you can see where the tumor is versus the stroma. Maybe you're interested in blood vessels. Maybe you'll have a phenotype that is dependent on the proximity of a blood vessel to a given cell type. It's also important to know the relative expression levels of each marker in order to properly assign channels. Large panels are probably going to include fluidigm antibodies in addition to fluidigm, or in addition to your own custom conjugations. And then we have free technical support available for help in designing the antibody panels. So it's not something you have to go at alone. We can certainly help you out with that and help you progress. So, what does the acquisition workflow look like? How do you select those regions of interest that you're interested in, and et cetera? So this is the software that we have. Here you can see that there's a region of interest outside of the panorama. This panorama is an image that is kind of generated with the optics inside the instrument, or you can put your panorama inside your ROI inside the panorama as well. You can work with, independent pathologists or really anyone that can help you identify the regions of interest. And you can actually annotate this on your slide ahead of time, right? So you can pick out those regions of interest. You can then ship your slide to the core at SickKids, and they can do a co-registration where they basically line the image that you sent to the tissue in the instrument and the image generated, by the instrument. And you can see here, we have this hole in the tissue. We have a very similar hole in the tissue here in the H&E. And we can line those up, and then where you specified ROI3, ROI3 is going to be there once everything's co-registered and your image is then ready to acquire. And of course, we can visualize the data as it's being acquired in real time, right? So I've sped this up, but you can see that the laser rasters across your tissue. And then if you look closely here, you can see that individual lines are being added to this image as the time goes by. Once you have your images collected, the next step is, a view and validate. This can be done in MCD Viewer, right? So here, this is software that will enable you to, visualize your images, make those pictures that you're going to use for publications. From there, you can send it off into Cell Profiler and HistoCat, or maybe VisioPharm and Halo, which are commercial solutions, to segment your cells and segment your images. And then from there, generally people will do some higher order analysis to identify differential expressions within a cell, between cells, the spatial context of cells, all of those kinds of things. Right, and here you can see, the cell profiler analysis, right? So you have your individual nuclei. People will then generally, extrapolate to where the membrane is, or have a membrane marker to extrapolate where the membrane is. And then from there, you can, save that single cell data for import into HistoCat, right? Inside HistoCat, you can, do that dimensional reduction and data analysis, identify different populations. And what's cool is you can identify populations on the right in your plots and in your graphs, and then highlight those back onto your image, which is a good, sanity check, among other things. And all of these solutions are open source and available to anyone to use for free. We also have some commercial partners, including Halo and VisioPharm. And with that, support and contact information, we can, you can reach out to Zishan if you have account questions, pricing questions. You can also reach out to me for any field application questions. And with that, I want to turn it back over to Sydney, because she has a few more points to make on, what the core at SickKids can offer. So thanks for your attention. And Sydney, if you want to pick it back up here.
Great. Great. Thanks so much, Eric, for that really informative and exciting presentation about the power of this technology. So I just want to very briefly come back to the service that we offer for those of you who might be interested. So at the moment, we're offering what we call a slide scanning service. So people who can stain their own slides with metal tagged antibodies. It's an hourly fee for the ablation time, and a setup fee because it can take some time to set up the instrument for the ROIs to be scanned. I put the approximate running times up for different tissue sizes. So in microns, so about 2 hours for a 1 millimeter squared or 1,000 micron area scan, and it goes up from there. And as Eric pointed out, we can set it up to acquire multiple ROIs from the same tissue section or from different sections on the same slide, and let it run for many hours at a time. And then lastly, I just want to come back to the antibodies. So as Eric said, we they offer a very comprehensive catalog of metal tagged antibodies, particularly for human, that have been verified to work on human tissue that's been fixed and paraffin embedded, so FFPE. I think there are fewer antibodies that they verify will work for mouse, either on FFPE or frozen. So we will offer a small panel of mouse antibodies that in our hands have worked on mouse frozen sections. So my slides are not advancing now. Oh, sorry, I didn't want to show that. Yeah, so here's just an example of some of our own data looking at mouse small intestine with Peyer's patch. And so we can see that there, we can stain T cells, structural markers like smooth muscle actin for lymphatic endothelium, epithelium with EpCAM, different T and myeloid markers and B cell markers. So we can get some really beautiful structural delineation of different cell types in different regions. And then lastly, we have performed a number of experiments with Fluidigm's pathologist verified antibodies on human FFPE. So on the left is a tonsil section that we stained, just showing you a couple of markers. And on the right is actually synovial tissue from an osteoarthritis patient. This is a collaboration we're doing with Kathy Simonovich to look at spatially at immune cells in various rheumatologic diseases involving the synovial tissue. So with that said, back to the antibodies. So just again, we can tag any antibodies for you if you can get them purified, or you can buy them pre-tagged from Fluidigm. So more questions about our service, you can contact Tina Chen. You can find her on our website for Casca, and she can give you more information. So thanks very much. I think we can open it up to questions now. Yeah.
Yeah. So thank you very much, Cindy and Eric, for a great talk, great slides, great coverage of the topic. Seeing that we have only 2 minutes remaining, we have had many questions come. No, listen, the topic was awesome, and the cool thing is that we do have a lot of questions coming in. They're all centering around cost, workflow, a lot of data analysis focused questions. So instead of rushing through the questions, what we'll do is, because I know we want to be very respectful of people's time, because I'm sure, even though many of us are, even working from home, it seems like we're working a lot more than we were working when pre-pandemic. So just in the essence of time, what we'll do is we'll take down all the questions, and we will be following up with all the attendees. So whatever we can share, whether that's the slide decks or at the very least the key questions answered, we'll share that with everybody. And as Eric and Cindy mentioned, there are resources such as Eric, Cindy, Tina, myself, that you can reach out to with any questions you have that maybe you did not get a chance to type in the question box or send to us previously, but we'll address all of them. So hopefully everybody got something valuable out of the slides today. Thank you very much for everyone's time, and we will be following up with answers to your questions.
Great. Thanks, everybody. Take care. Bye-bye.